All right, we're going to get started. Welcome, everybody. I know there's a lot of people joining, so I'll talk a little slow. But today we have Simulations Plus, and this will be a panelist slash roundtable discussion with Q&A at the end, and also a short talk by Timothy McGovern. And the title is Beyond the Lab, FDA's Vision for Modeling a Future Without Animal Testing. This is a long time coming for many of us who have been in this industry for, 30, 40 years. And it's a very welcomed evolution. So today's agenda will be just the rules of engagement, very simple, nothing crazy. And then a presentation by Tim McGovern, and then a panel discussion moderated by Sandra Suarez. And then we'll go into Q&A at the end. It's going to be a packed time, so we're going to move on here. So the rules of engagement: put questions or comments in your chat or QA box at any time. We will get those answered at the end. If for some reason we run out of time, we will follow up with you with those answers to those questions. We typically answer all of them. The chat box, people can see your name. The Q&A box, it will be anonymous. So if you don't want your name seen by others, then follow those directions. Again, we're going to try to address all questions. So far, we've addressed all questions in the past 5 years. Everyone will have access to the recording of this afterwards. So that's the important part. If you have to leave early, get your questions in there, and then you can come back and watch the recording. So let's just do introductions for everybody. So Sandra Suarez Sharp is the Vice President of Regulatory Affairs at Simulations Plus, where she leads the Regulatory Strategies Center of Excellence. She brings over 2 decades of FDA experience, specializing in biopharmaceutics, PBBM, and model-informed drug development. A former scientific advisor in the FDA's Office of Product Quality, she contributed to major guidances and scientific initiatives. She has a PhD in pharmaceutical science from the University of Florida and has made significant contributions to regulatory science through publications and leadership. Vera Lukakova is the Chief Science Officer at Simulations Plus. She's involved with GastroPlus, DDD Plus, and Membrane Plus software packages. She has contributed to drug development modeling studies, interactions with regulatory agencies. Scott Seiler is the Chief Scientific Officer of Quantitative Systems Pharmacology at Simulations Plus. He leads QSP and QST efforts and has significantly contributed to NAFLD SIM, IPF SIM, and DILI SIM. Scott has had major involvement in investigational marketed drugs dealing with metabolism and liver diseases throughout his career. Previously, he spent 12 years in the industry working on metabolic modeling projects. He holds a PhD in nutrition from UC Berkeley and a BA in biochemistry and molecular biology from UC Santa Cruz. Susie Zhang is an expert in regulatory science and pharmaceutical development with experience in both the FDA and industry. At the FDA, she was recognized authority in PBPK modeling and model-informed drug development, contributing to key regulatory guidances. Her expertise spans biopharmaceuticals, clinical pharmacology, PK/PD, DDI, and modeling with around 70 publications. She has supported diverse programs at Daiichi Sankyo and holds a PhD in pharmaceutical sciences from the University of Michigan. And Tim McGovern is a co-founder and principal consultant at White Oak Regulatory Toxicity, where he provides regulatory advice and support for all aspects of non-clinical drug development. He recently retired from the FDA, where he was an associate director for pharmacology, toxicology in the Office of New Drugs. He's an active in policy and guidance development, non-clinical and regulatory issues, including FDA and International Council for Harmonization initiatives on topics including carcinogenicity, assessment and assessment of DNA reactive and elemental impurities. His formal training is in the field of inhalation toxicology and began his career at FDA evaluating safety of pulmonary and allergy drug products. And with that, I'm going to turn it over to Tim, and he's going to give a short talk here. And I'm going to do the slides. Okay.
Great.
Tim, you tell me when to advance the slides.
Will do. Thank you, John. Thanks, John, and all the organizers of the webinar for inviting me to participate. As John mentioned, I'll provide a very brief introduction to the issue to help set the stage for the panel discussion. And next slide. So first, I just wanted to cover, what do traditional non-clinical testing programs look like? And recommendations are provided in ICH guidances M3R2 for small molecules and S6R1 for biologics. In general, the types of studies conducted are described on this slide, and they can include a mix of in vitro and in vivo studies, especially for pharmacology, safety pharmacology, PK/TK, and genetic toxicology. Additionally, weight of evidence assessments can be provided to support carcinogenicity and reproductive toxicology assessments in lieu of some animal studies in certain cases. Next slide. So in April 2025, as I'm sure many of you know, the FDA released an announcement of a plan to phase out animal testing requirements for monoclonal antibodies and other drugs. The announcement refers to the plan as a groundbreaking step to advance public health by replacing animal testing in the development of monoclonal antibody therapies and other drugs with more effective human relevant models. It goes on to say that the animal testing requirement will be reduced, refined, or potentially replaced using a range of approaches using new approach methodologies or NAMs. The announcement, led many to believe or to at least ask if now animal studies were no longer needed to support monoclonal antibody development programs. I've heard that question, a number of other consultants I've interacted with, CROs as well. That's not what the announcement and supporting documents say. Certainly that's the eventual goal, but not quite where we're at right now. So next slide. And so I just highlight one part of the announcement. It refers to a provided and provides a link to a roadmap as to how this plan would be implemented. And next slide. And so you're looking at the roadmap itself. The implementation plan is titled Roadmap to Reducing Animal Testing in Preclinical Safety Studies. And the roadmap outlines a strategic stepwise approach for FDA to again reduce animal testing in preclinical safety studies with scientifically validated NAMs. Next slide. And so, a few high-level comments on the roadmap. The program is intended to begin again, with monoclonal antibodies as a promising area for reducing animal use. It would eventually expand to other biological molecules and then eventually, further on, new chemical entities and medical countermeasures. A long-term, 3 to 5 year timeline, long-term vision would be, no conventional animal testing will be required for monoclonal antibodies, safety testing, and as I said, eventually expand to all drugs and therapeutics. And it states that FDA aims to make animal studies the exception rather than the rule, and that animal studies might only be considered if a specific question cannot be answered by a NAM. So that's the long-term vision, again, with the primary focus on monoclonal antibodies at the moment. So next slide. The roadmap describes the various NAMs that have been under development over the years and could be considered for use. And they are listed here. They include in vitro human-derived systems, in silico tools and computational modeling. A number of examples are provided. It indicates, as part of that, these tools may act as powerful adjuncts or replacements for animal studies, and that thresholds will need to be developed or modified for when animal testing can be reduced or eliminated, and those should be continually updated as we get more information. And it also talks about other innovative platforms that can be taken into consideration or developed. Next slide. This slide includes a list of some key considerations pulled from the roadmap that I thought were important to highlight and will likely come up as part of our panel discussion. And I'll further highlight a few here. One is, the submission of NAMS data to FDA. This has been an ongoing request, from my time at FDA and, still continues, encouraging industry as they're generating this type of data to submit it to the agency for evaluation and consideration. Um, Another one, establishing the validation and qualification pathways, very important. Creating a clear context of use, and I'll talk about that more in a minute. Use of an integrative strategy. So, the question of, particularly depending on what the question is, one approach may not be fully adequate. So integrating, a number of different approaches can be useful in certain scenarios. Developing or updating FDA or ICH guidances. For example, if FDA updates a guidance, but other regulators are still asking for animal studies, then in all likelihood, based on global development, those animal studies would still be conducted. Um, Encouraging of, continuing training and communication. There's been certainly a good amount of that from my experience over the years, and that should certainly continue. And finally, it states that FDA must assure that any new method is equal or superior to the animal test it replaces in protecting patients. So I think that's a key consideration. Next slide. And I just wanted to note that this proposal is not completely novel, but really builds on previous proposals and work that has been ongoing for quite a while. One example that I'm putting out on this slide is that in 2017, FDA released an FDA predictive toxicology roadmap, which talked about developing many of these same tools and moving the area of toxicology forward from a firm reliance on animal studies to where some of these new technologies, could be developed and eventually augment, gradually and gradually replace animal studies. Next slide. And this just a website hosted by FDA, focused on alternative methods that provides, I think, useful information on various aspects of NAM development and regulatory application. Has all kind literature references out there, different meetings and workshops that have been developed over the years. So I think that's a very helpful resource to take a look at. Next slide. And I wanted to highlight two publications from former FDA colleagues in the Office of New Drugs in CDER that provide regulatory perspectives, identifies key areas where NAMs can be applied and where some of the knowledge gaps, have existed, particularly over the recent years. Next slide. And one issue I wanted to point out, getting back to that issue of concept of use, really one particularly important issue that was mentioned in the current FDA announcement, and this issue is addressed in the Avila et al. 2020 paper, and talks about context of use refers to a clearly articulated description delineating the manner and purpose of use for a particular approach. And having a clearly defined context of use makes it easier for all involved to envision, the potential value of that approach and helps define the amount of information needed to adequately qualify the approach. And so I think it's important to note that the amount of information needed to support the use of a NAM to address a very specific, for example, potentially adverse finding from an animal study is likely going to be quite different than that needed to replace an entire study or set of in vivo studies. So really, context of use matters, a great deal. Next slide, please. And then finally, I just wanted to mention the FDA Modernization Act of 2021 is also known as the Modernization Act 2.0. As noted on the slide in the Federal Food, Drug, and Cosmetic Act, the term non-clinical tests or studies replaced animal, and the act clarified that approaches other than animal studies could also be used to assess non-clinical safety of pharmaceuticals. When this was passed, it caused a stir, somewhat similar to the April announcement, with many thinking that animal studies were no longer needed to support the safety of drug development programs. But in reality, it was really more of a clarification of the actual practice than of a change in regulatory position, where again, these approaches other than animal tests can be used as part of the overall regulatory submission package. Currently more as complementary or supportive in terms of addressing a specific issue, but again, with the goal to eventually, you replace some, if not, further down the road, all animal test study, all animal-based studies. So with that, I thank you for your attention, and I will turn it over to Sandra for the panel discussion.
Yes, thanks, Tim, for a very nice background. And as you said, it's important to keep in mind that the FDA announcement back in April follows years of advocacy, including the FDA Modernization Act that was signed in 2020. With that, I'd like to invite all the panelists to join us for the discussion. Just to start, we have prepared several questions that are very relevant to providing further clarification of the agency's expectations regarding the phasing out of animal testing and a path forward addressing the agency's recommendations. So with that, let's start with the first question. With recent commitments and communications from the FDA and EMA to reduce and phase out animal testing, what changes or new approaches will the industry need to adopt moving forward? And can you also comment on what are key challenges the researchers and developers encounter when implementing NAMs? Even more so, how might these changes impact the timeline and cost of drug development? And I'd like to invite Scott to start with the discussion. Scott?
Thank you, Sandra. I'm super excited about that. Everyone has acknowledged the likelihood, the possibility, the potential for reducing animal studies. This is a fantastic development, and clearly lots of internal, external momentum has been provided there. So super excited about that across the board. Also, in science, you're always excited about new methods, right? And new methods open the door, unlock the possibilities of learning more. And in this case, we get a perfect confluence of the two: learn more and replace something that is perhaps not necessary and comes with some liabilities. So that, just generally speaking, that's fantastic. Now, some of what we're calling NAMs are not all that new. And of course, I'll talk about mathematical modeling. Decades of use of mathematical modeling, increasingly in the pharmaceutical industry, increasingly accepted by regulators. And so in some ways, we're calling it NAMs, but in reality, what we're saying is we've learned these methods, we've relied on these methods, there's been some validation of them, and we're able to now really truly use them. On the other hand, there are new methods that are truly new, just now arriving on the scene. And so we're kind of balancing both of those. I like the description that Tim gave, where there's sort of a ramp, right? Where we'll be dialing down our in vivo studies and the magnitude, the number of them, as we ramp up and onboard these NAMs. And so as long as all of those come together, there will be time for that, there'll be effort for that. Once they've been implemented, though, not only do the NAMs have the potential to replace and reduce animal testing, in reality, they'll likely accelerate the preclinical side of drug development as well. And so to me, that's just a win across the board.
Thank you very much, Scott. And you mentioned some very interesting points, and you talk about the tools model not being not being a new approach for supporting this effort, and also about validation. We will definitely touch upon this a little bit more in further discussion. I'd like to invite this time Tim to also give his input on these questions.
Yeah, thanks, Sandra. Yeah, I agree. I mean, I think everyone involved in drug development is looking for ways to reduce and eventually replace animal testing with the right tools in place. And as noted, some of these approaches have been widely accepted for years. Things like QSAR assessment for DNA reactive impurities, the Ames assay for mutagenicity assessment of APIs and impurities has been around for a long time. That was really the original NAM in my mind, or one of the original NAMs. Other ones, like I'd mentioned, for early prediction or weight of evidence approaches for supporting carcinogenicity assessments. More recently, more recent approaches that have been put into place and have been so far, fairly successful. Also, industry uses many of these approaches already, early in development to support lead candidate selection, and this type of information is often not submitted to regulators for various reasons. And I think so. There's a lot of experience on the industry side, kind of looking at these approaches, and it just has not translated to a great degree over onto the regulatory side in support of actual submissions. Development of organoid and MPS has been rapid and evolving. And I've interacted with the NCATS program at NIH over the years, and just the engineering behind it is amazing. But one issue is that it's constantly changing as these technologies are increasing, and so getting into the whole qualification and validation issue in terms of what is the actual system you're looking at, and as changes are made, how representative is the data generated with the previous version with the newer version. So I think that's an issue that needs to be addressed as well. And I think people are addressing that as we move forward. I think there's certainly a need to continue outreach to regulators, across regulatory and industry representatives to increase awareness of and experience with new models. It's important to be objective and realistic regarding what a given model can and can't do and what it's really being applied for. Consider how various models can best be integrated. That was one thing mentioned in the FDA announcement, is that one model is likely not going to address an issue, and that it's likely going to involve integration of multiple models. And then the end question being how many systems will be needed to adequately recapitulate in vivo studies with that end goal coming in mind. A question is how best to mimic PK and the drug in vivo and confidently estimate human doses and exposures, how to assess consequences of long-term exposure, which can be difficult in an in vitro system. And then, like I said, submitting data for regulatory evaluation. And so those are, I think, some of the challenges, just getting everybody on board. In terms of cost and timeline, what I guess first question to come to mind is really what percentage does non-clinical safety and efficacy testing really comprise for the overall development program? And in general, it's a small portion overall. Initially the timeline, Scott had touched on this, more of a kind of a ramping up of these approaches in combination with animal studies. So I think initially the timeline will not be significantly impacted, as you're going to be you're probably seeing NAMs submitted along with standard studies, and there could actually be some increase in cost as you're generating additional data. But eventually NAMs could replace some animal studies, reducing the overall cost. But in terms of, certainly timelines for the non-clinical data could be reduced, but how that impacts the overall drug development program to approval, I think is debatable, because again, non-clinical is one aspect of the overall development program. So, thanks.
Thank you very much, Scott and Tim, for your input. And I think we're going to move to other questions we have, because we have several. But just to summarize, we heard that initial cost and timelines may not change significantly, but long term, I would think that the use of validated NAMs could improve efficiency, right? And for this to happen, industry must adapt by integrating new methods, validating models, and engage with regulators as early as possible. And in this context, Piera, could you please mention what are Simulations Plus plans to support NAMs to reduce reliance on animal testing? Specifically, what ongoing initiatives at Simulations Plus are aimed at expanding the NAMs portfolio to further support this transition?
So I'd say what our plans are is keep doing what we've been doing for over 20 years now. This is exactly what we've been supporting. We just need to accelerate and keep building these models and bringing them to our users and to the industry even faster. And with a bigger focus also on the biologics. Right? I'm sure that we have here on the call, many of those of you who are listening to us have been working with us, especially in the small molecules, right? Working on using the modeling and simulation We may not have always talked about it, that this is to reduce the animal studies, right? We've been talking about it. This is to improve the first in human predictions. This is used to improve other types of extrapolations. But what it ultimately leads to, when you are using your data to the best of your ability, right? When you are utilizing all of the data that's available, it ultimately is going to lead to having to do fewer experiments, especially fewer experiments in the animals. So going forward, the plans are really continuing with the collaborations and focusing the developments on the tools that are ultimately leading to these improvements and to minimizing the animal use through better use of the data that is already generated, right? And again, focusing more on also the biologics rather than just the small molecules.
Thank you, Vera. Very interesting. I'd like to invite Scott to give his perspective from QSP and QST.
Yeah, again, I completely agree with everything, excuse me, that Vera just said. I'd like to also minimize the frog in the throat effect, if at all possible. But one of the things that I think I really want to highlight, two things in particular. One is, just to reiterate what Vera said, we're not only focused here on monoclonal antibodies at Simulations Plus, we're focused on all molecular entities, right? And evaluating them from exposure, from toxicology, from efficacy points of view with our mathematical modeling. And so we've been doing all of the above across all those modalities for many years now, decades, truly. However, as we look forward, and specifically to the first step of that roadmap, the monoclonal antibodies, one, I think, really interesting next step for us, right around the corner, essentially, is a model of what we call quantitative systems toxicology. We call it biologics sim, where it builds on our efforts in the drug-induced liver injury area with small molecules, and now enables us to predictively begin to evaluate how biologics may incur liver toxicity issues. We're even taking this a step further. More exciting to me is it's mathematical modeling combined with some NAM in vitro data collection approaches. So really, we're minimizing altogether the animal studies on the safety side, on the efficacy side, whether small molecules, whether biologics, and really enhancing our portfolio, our options to be able to do so.
Thank you, Scott. As Simulations Plus continues to lead in reducing animal testing through its, Vera mentioned, model-first approach, and you also mentioned, Scott, mechanistic modeling in areas such as QST and QSP. Scott, could you please elaborate more into what unique advantages does mechanistic model offer compared to artificial intelligence? Can these two approaches be complementary? And also, what do you think, in which ways, or what ways can computational models and organoids enhance the predictability of human responses compared to traditional animal models? So we are comparing here mechanistic models versus artificial intelligence and artificial models versus traditional animal models. Could you please comment on that, Scott?
Absolutely. Again, trained scientists here, all methodologies, where first thing we learn have their strengths and their limitations. And that really is one of the things that differentiate them and illuminates really kind of what the conclusions are, right? The limitation of one method means you can only deduce so much from it. And mechanistic mathematical models that represent human physiology and pathophysiology allow us to make predictions within the context of that in vivo environment. AI often, right, so requires a ton of data to be done properly and allows us to really determine connections within those data sets. So to some extent, I believe, Vera and I concur on this. AI really allows us to interpolate with great confidence, whereas mechanistic modeling allows us to extrapolate beyond that. It's also, I think, reasonable to consider that it isn't an or scenario. It can also be and. One of the things that we do at Simulations Plus is to combine AI approaches and mechanistic modeling, whether it's QSP, QST, or PBPK, in order to leverage the greatest aspects of each. And so, together, I think that's probably the future, right? Is to combine these things rather than to have them compete with each other.
Great. Then let's hear from Viera. Maybe she has a different perspective from PBPK modeling.
Well, now it's my time to agree with what Scott said. Yeah, I absolutely agree with we need to be using all the tools that are available, right? And each of them has its advantages. Again, from what I've seen and explorations, I would agree that the great power of the AI is that it can quickly sift through all of the data that's already available, right? Huge amounts of data have been generated through years and years of developments and research, but it will have hard time going beyond of what it has seen, right? It can recognize the patterns in the data that it has seen, but the extrapolation may be more difficult, right? There is a quick summary of what the AI means that somebody sent me just last week, that treat AI agents as new class of junior employees, right? They are cheap, fast, prone to error, and in need of supervision, right? So that's where they can really help, but we still need those mechanistic models to supervise and to help and allow the extrapolation. And in those terms, also going from the new in vitro assays, right? The organ on a chip, right? The microphysiological systems, we are measuring in vitro data in systems that are more relevant for the human, right? But at the end of the day, it's still just a small slice of the whole physiology. So we still need these mechanistic models to translate what we've measured in those systems into what will happen into the whole human, right? So all of these models do have that ability and to help with those, getting us the best out of all the information that is being generated and that has been generated in the past.
Thank you, Viera. So if I were to summarize what you and Scott mentioned, mechanistic models shine in extrapolating to new scenarios by embedding compound data with full biological and physiological context. But there are still some questions, I think, around scalability and how many systems are needed to replicate in vivo complexity. So this leads us to our next question, which is critical for practical implementation, right? We have heard about validation over and over. And so this specific question is about regulatory acceptance and qualification of NAMs. So specifically, how are NAMs being validated and accepted within the regulatory frameworks? Maybe you give from FDA perspective, global perspective. Please elaborate, and let's start with Viera.
Right. So where they are being used, the advice, and this is a question that I often get asked, right? From the scientists utilizing these models and so on, how can we ensure that the model that we have is going to be accepted, right? The answer is always the same. You need to show the reviewers that you know what you are doing with the model, right? It comes down to the validation. The model needs to be validated for purpose, right? T2 is to show that the model is really able to predict and to answer the question that you are trying to use it for. Right? So you need to show the solid science behind the model, solid understanding of why the model is being used, right? And that the model is able to predict the scenarios that you are looking at. So at the end, it comes down to if you can put a good, solid scientific explanation behind the model, right? And show that it's predictive, it does have a good chance to be accepted in the regulatory applications.
Thank you, Viera. So this is a perspective from a modeler's mind. So let's hear about from people with regulatory background, and I'll invite Susie to give her perspective.
All right, thanks, Sandra. I think we're talking about, Viera talked about model validation. We heard about model validation a lot in the past. Myself, background is modeling simulation, but today we are talking about NAMS, which is broader than modeling simulation. It includes organ on chip, how those systems are validated, microphysiology systems, cellular systems. Those are even broader than modeling simulation. Initially, I think this type of validation should be or would be case by case. Recent guidances have been talking about context of use. I know FDA likes this phrase, context of use. You've heard about it even from today's presentation a few times. But if you look at guidances across different disciplines, including farm talks, modeling simulation, it's been there in a few guidance. For example, this year's AI guidance and also this year's M15 ICH MIDD guidance. They all use context of use, which is closely associated how you are going to develop your validation plan, whether it's a model validation or it's an in vitro system validation. But I think the principle behind it is similar. It's scientifically sound, it's relevant. You use all the data you have available there to set your validation criteria. And also in all those guidances, it has been mentioned that early engagement between developers and regulatory agencies are encouraged. So that means at initial stage, maybe agency is still exploring the same as our developers. So early engagement and communication between those parties are important so that we can align on how those model or the in vitro approach should be validated. And if we review the recently published new approach methodology cases that have been accepted by the agency, I noticed there are a few commonalities. For example, the targets are previously studied with clinical experience. And also another important aspect is that one approach may not be enough. You probably need to have a combination of approaches. And in silico is often one of them because it's a tool that allows you to link your in vitro data, animal data, human data all together. And I would say it's an integrated tool that can build your knowledge and then do predictions. I think as laid out in FDA's roadmap, recently published in April, it appears that in the next 3 to 5 years, validation will be one of their focus areas to work on. I hope that helps.
Oh, definitely great to see. And just because I believe this is one of the major issues or things that we need to focus as scientists. I like to hear from Tim, his perspective. Tim.
Yeah, thanks, Sandra. So to answer that question, how are they being accepted? How are NAMs being accepted within the regulatory framework? I think at least on the drug development side, slowly. More so, I think in areas like in the environmental fields, food safety, even tobacco, where they're being incorporated more readily at this time. And those are areas, particularly in the environmental side, where there's a larger set of chemicals to be evaluated, and we're testing all of those chemicals, through empirical studies, may not be feasible. So that's helped to move this area forward in the NAMS field. I think the recent FDA announcement, one of the likely aims is to speed up that process for pharmaceuticals. So I think you'll probably see a lot more attention on that in that area. Again, and as mentioned, just by Susie, again, identifying that context of use is critical, and providing that data to support it. Again, that the statement in the FDA announcement assuring that any new method is equal or superior to the animal tested replaces in protecting patients. And then identifying and communicating, as Susie was talking about, successful case studies where NAMs were accepted, and what that type of integrated assessment might look like can be informative in terms of supporting, approaches moving forward, particularly in being accepted in lieu of in vivo studies. And then, moving forward and, that formal incorporation into regulatory guidance with clear expectations, and incorporation in guidance does not guarantee, immediate implementation by industry either. I recently gave a talk at a course for the American College of Toxicology on carcinogenicity assessment, and in 1996 or so, the guidance, the S2, no, the S1A guidance, S1B, introduced the ability to conduct 6-month transgenic studies for carcinogenicity testing. But in looking at some publications, even not a simple change, but a relatively isolated, focused change in non-clinical development program, it took about 10 years before industry was really using that type of a shorter duration study, less expensive, and really felt confident enough to start using it in lieu of the standard 2-year study. So, just making the point, just because it gets into guidance doesn't mean industry will take it up and move forward, right away. Obviously, that's the goal, but you always have to wait to see what happens.
Great, thank you so much, Tim. Just to summarize, based on the input from our panelists, NAMs are evaluated case by case, requiring strong validation through combined approaches and proof that they match or exceed animal tests in protecting the patient. We heard from both Tim and Susie that history of use in scientific or regulatory settings is key for wider acceptance. So in this context, Susie, could you please talk about what does the future hold for the expansion and global harmonization of NAMs in regulatory science? Specifically, what has been the FDA's experience in reviewing NAMs and how this has shaped the path forward for integration of non-animal testing methods?
Yeah, so when we look at a few publications or a few accepted NAM examples in the published domain, they were all submitted to different agencies and reviewed by different agencies. Of course, those are case by case. I'm hoping that in the future that it's also in FDA's roadmap white paper where they're going to take initiative to harmonize this NAMs. I'm hoping that the ICH guidance will be worked on and could be faster than before based on the few recent ICH guidance that I've seen. It seems that different regions and agencies work accelerated steps to get this global harmonizations done. This is important because if we go back and think about the roles of reducing NAMs, it's to reduce unnecessary studies. It's to develop the drugs in a scientifically sound way. However, if different regions have different requirements, then the developers will think, does it make sense for me to submit different packages to different regions? It still takes time, cost more. Well, maybe cost more if we have to submit different packages. So that doesn't serve the original purpose of doing this NAMs. But I'm hoping that FDA and also other regulatory agencies will work together to get this harmonization guidance out as soon as we accumulate enough experience as well, case examples.
Thank you, Susie. Tim, so tell us what's the FDA experience has been on NAMs.
Yeah, I think Susie covered a lot of it. I think a lot. When you're talking about global harmonization, you're really talking about the ICH guidance development. Certainly FDA has stated its position that this is a priority issue and that they'll be pushing forward in guidance development, at least on the domestic side, and will be making this a high priority in terms of ICH harmonization as well. Because again, that's really where the real value is if you have harmonization across regions, given global drug development. So I think harmonization will continue to grow as focus increases on this. Europe in particular has been very active in this area to reduce animal testing, particularly with the REACH initiatives. So I think you're going to continue to see increased focus in this area. It's going to require additional data generation to support more widespread use out of very specific outside of very specific applications where yeah, at least in terms of, non-clinical assessment with organoids and MPS systems have been used to date. And like I said, FDA experience on the drug side industry has been relatively limited based on, lack of, significant amounts of data submitted. But also internally, FDA researchers have been active in engaging with outside developers to further enhance the regulatory experience and integration into the regulatory framework. So I anticipate those types of partnerships will continue. Obviously, there are issues regarding staffing and funding across the federal government right now. So what role all of that impacts moving this type of approach forward in a streamlined, efficient fashion. I think that remains to be seen as well. But certainly there are ongoing efforts to try to achieve the goal.
Great. So if I were to summarize very briefly, we heard that if the experience in evaluating NAMs is rather limited, but despite of these regulatory agencies, EMA, FDA have encouraged their use because I think that they understand its value. And obviously, as NAM gain traction at the FDA and globally, it's clear that adoption won't happen in isolation, and we all need to be part of this effort. And so we have one question left that we have prepared, but I think we have like 10 minutes left in our schedule. So I think that we're gonna at this time turn to the questions being added to the chat, and let's go to it. Okay, so let's see. The first one. The initial focus seems to be on antibodies. What are the specific challenges for NCE? What is already available for NCE? Anybody? So I think can you see your question in the chat one more time? The initial focus seems to be on antibodies. What are the specific challenges for NCE, and what is already available for NCE? New. I say NCE most likely be new
chemical entity.
New chemical, yeah.
Yeah, I can jump in with an initial thought anyway. I think monoclonal antibodies were identified as kind of the lower hanging fruit to really focus on for an initial approach, just because number one, it's a biologic. There's less off-target potential effects that you're going to run into. We have relatively good history of experience with monoclonal antibodies. Some are some are less complicated. There are still some complexities with some areas in the monoclonal antibody field, but I think that was at least looked at as an initial area of relatively low concern to start incorporating these types of approaches in place. So I think that's why it started. When you're talking about more of the small molecules, you have greater potential for off-target toxicities and more widespread toxicity profile. That again, it gets to that question: how many models do you really need to adequately address potential concerns? So I think that was really one of the drivers anyway for focusing on monoclonal antibodies at the start.
Thank you, Tim. How you want to elaborate on this question, Scott and Viera, because you already said this is not new. Please.
Viera, you go first, and then I'll take it from you.
I'll get started with the PK side, right? This is where actually, when it comes for the small molecules, that's where we have more developed models on the mechanisms, right? And going from in vitro to in vivo predictions and so on. Depending on what kind of compound you are working with, we may be able to already avoid animal studies, but we definitely are at the stage where we can start reducing them, right? That's where the models are better developed. And we do have a lot of those mechanisms especially on the PK side, to start predicting the distribution from the in vitro data, or from smaller number of animal studies and doing those extrapolations. I'm sure Scott has more on the toxicology side, especially.
Yeah, so with both predicting efficacy and toxicity, that's the first step is making use of what Viera just described, right? Making sure the exposure at the site of the target is properly considered. And new chemical entities or small molecules, however you want to describe it, tons of tools available to do that, PBPK modeling being at the forefront of that. I think we could talk for hours at this, but I'll give one specific example of a great tool that is accepted by regulatory agencies. And it's in the drug-induced liver injury space, DILIsym, it's called. It is actually the liver safety reviewers at FDA have it on the accepted non-clinical list for ways of evaluating toxicity risk when it comes to liver injury. This is a fantastic development, right? It's exactly what we're talking about, is the ability to use NAMs, in this case, mechanistic or QST modeling, to make predictions to support ultimately making sure that drugs are safe. Fantastic.
Great. Susie, you want to comment on this from a case study perspective, or you think it's already covered? We can go to next question.
Completely agree with Vera and Scott and Tim. Everybody said we're definitely at the reducing side for new molecular entities, especially small molecules. When we were talking about NCEs, reducing, we have to think about the studies that are relevant. Everybody has limited resource, and we wanted to optimize them. Which studies can be limited or even reduced the length of the study, or incorporate some of the clinical monitoring in the clinical programs when the new molecular entity goes into clinical stage. Those type of things should be considered.
Thank you, Susie. Let's go to another very interesting question. This is a very good step forward, as pointed out by Tim and Scott. However, many times humans are naturally worried when big new changes seem to be coming. Looking from the side of less developed countries, where even animal testing is not established in some countries, this will definitely stifle their efforts in drug development. Will there be mechanisms to ensure these new systems are not extremely expensive? Scott.
What typically happens, right, is the cost is larger at the beginning. Then as we become more efficient understanding, supply chain, all of that starts to kind of subside those pressures, and we arrive at a lower price point, a lower cost for doing these studies. Already, in some cases, NAMs have followed that paradigm. I anticipate the same. So there will be some cost burden, that is certain, but it should settle at a place that hopefully is feasible for all.
Great. Vera, you wanna comment on this?
No, the only thing I can add to it that in this transition phase where we are, right, where we are still doing animal studies and doing also modeling, right? It may seem like more expensive, as Scott and Tim were also mentioning earlier, but over time the costs for animals are going to disappear, right? So, and in computer, you are not limited how many studies or how many animals you are going to do, right? You can do 1,000 simulations, right, for the cost of one simulation. So
yes,
I'm really puzzled by long term. The cost should be going down once we are switching more into the computer and the in vitro assays, eliminating the animal studies.
Thank you, Vera. Let's move to another question. Especially in the field of oncology, we sometimes have limited clinical data in patients. How should modelers handle this when they want to focus on modeling, but they need to verify their model with limited data sets? Any thoughts, recommendations? Let's see who wants to start with this one.
The focus on modeling, the validation, right, and evaluation of the model is part of the modeling. Right? So there is, it's always integral part of it. It's not either or, right? That I'm either developing the model or doing verification of the model, right? The verification of the model is part of building the model. So there is no way to separate those. The limited data sets, right, that brings uncertainty. So that is the aspect that always we are dealing with if there is limited data for validation. But this is where, again, over time, as we are improving the models, the historical evidence starts coming in, and perhaps the requirements on the validation will be lower, right? If, right now you might be asked to do validation with many different studies. As we are all developing and getting that confidence that these types of models really work, right? The requirements for validation should also go down because that historical evidence is already there.
Yeah, maybe I can
Susie, yes.
add a little bit that. I wouldn't think about this as oncology, but more like towards rare diseases where clinical data is always limited. We don't have a lot of understanding or good understanding of the disease progress. In rare disease area, how the modeling simulations are doing, it's actually more important than the other areas because modeling simulation is a tool that helps you to connect all the dots together from in vitro, clinical, preclinical, historic data, and everything together. I wouldn't think more from modeling validation perspective, but think about it as a tool. All kinds of data available, even indirect evidence. Can we use that? So I think that's a continuous challenge, not only for oncology, but a lot of the rare disease areas. And we're using more and more of these innovative tools to develop drugs treating those diseases as well.
Thank you, Susie. And I think all that is, I would agree, is very well known. For years, we have been using historical data. Agencies, specifically EMA, has relied on articles, published literature to qualify, validate, to gain confidence in certain application for the model. Tim, I don't know if you want to elaborate beyond mechanistic modeling when it comes to validation of in vitro testing. You have any experience on that?
No, I don't. So,
It's okay.
But I think what the previous comments made a lot of sense.
Wonderful. Yes. Okay. Let's move to another question. How will the acceptance of NAMs be coordinated across ICH regions? Will we still be doing animal studies to support global submissions?
I guess I can jump in there, I have a good deal of ICH experience anyway. So currently, yes, you'll still be doing animal studies to support both, I think, domestic and global submissions. Generally, one of the ICH parties will propose, a new guidance topic or an update to current guidance. So, either approach, I think the FDA roadmap kind of talked about potential for either of those approaches. There is wording typically in guidances that talks about, what the recommended approaches are, but that alternate approaches can be used, with sufficient justification. So I think, the FDA roadmap is talking about, putting more specific kind of meat on those bones for that alternate approaches for, specific to NAMs. And I think, certainly I think other regions are well aware of these approaches going on. But moving it through the ICH process, has at least one party kind of proposing a topic, whether it's a new guidance or an update, and then the other parties, essentially agreeing to reopen an already existing guidance or start a new guidance. And so oftentimes there are competing priorities as part of that. Where do NAMs fit in terms of all of the other proposals coming forward? What the bandwidth of, for the various ICH parties to take on new guidance development. So I think, over time, this is obviously a growing area of awareness and emphasis. So I think over time there will be that consensus generated. I don't know when that will be, but probably not in the too distant future to start working some of this into ICH guidance. And at that point, when those guidances are finalized, those updates are finalized, then certainly you would achieve some global harmonization. But that doesn't mean these types of approaches cannot already be submitted to regulatory applications to support a particular rationale to address a given safety issue that might turn up. Or as we've talked about, there are already a number of these models already well in use in particular situations in drug development. So thanks.
Thank you so much, Tim. In the interest of time, perhaps we could take only one more question quickly, a minute response. In many cases, and this is from Brett Hall. Hi, Brett. In many cases, animals are terrible predictors of human outcomes, and we have all recognized this for years. However, when NAMs come up, many people immediately focus on their predictive power compared to human outcomes, as opposed to comparing them to the current standards, which is not very predictive animal studies. Question: what are your perspectives on shifting the regulatory and industry focus towards NAMs as better than animals, not perfect models? Who wants to take this? Scott, Viera?
I can start, since this is something that, again, in PBPK world, we went through 20 years ago when trying to move from the typical traditional approaches of allometric scaling, right, into more PBPK. The focus there was exactly as Brett outlined, right? It wasn't about, can the PBPK do a prediction within these limits, right? It was, can the PBPK give us better predictions and more reliable predictions than the traditional allometric scaling, right? And so I completely agree. That's where the focus needs to be. It's not putting in some standard or predictability against human data. It's evaluating all the methods together and selecting or moving forward with the one that gives better predictive power, even though it's not perfect. But why wouldn't you use the one that's giving you more predictive power, even though it's not perfect, than the one that's even worse, just because it's been used for years?
Yeah. Well, thank you, Vera, for very nice insight, and everyone. With that, I'd like to thank everyone for their participation and interest and your very insightful questions. To reiterate, we will address the remaining questions at a later time. Please let us know if you can contact you. And just to conclude this webinar, let me give you some take-home messages, some sort of summary, right? Take me a minute or two. We heard that mechanistic modeling has unique strengths in improving the predictability of human responses, which will reduce reliance on animal testing. And modeling, such as PBPK especially, has been used in this context for a while. So we have regulators have confidence in this kind of mechanistic modeling. To improve or increase the likelihood of regulatory acceptance, models should be validated within a clearly defined context of use. We heard this over and over by Susie and Tim. Ideally, by integrating multiple complementary approaches, such as in silico, in vitro, and in vivo methods. To advance NAMs as credible and widely accepted alternatives to animal testing, regulatory agencies must gain confidence in these approaches, right? So it will depend on transparent data sharing. In other words, we need to be sharing that data. Also, cross-sector collaboration is going to be very important, and training and tools and methodologies will be important, very important, because in turn it will be converted into regulatory guidance. Lastly, the FDA aims to make animal testing the exception, not the norm, to predict safety testing within the next 3 to 5 years, perhaps a little longer, you never know. This shift emphasizes the importance of becoming familiar with NAMs now, not just for monoclonal antibodies, but also for biologics and small molecules. So I really exhort you to let's prepare, because this transition, if we are prepared, this transition will ensure that we remain aligned with the regulatory expectations and contribute to the development of safer and more cost-effective therapies for patients. Thanks again for attending today, and hopefully we get to engage in another discussion when it comes to NAMs. Have a nice rest of your day, everyone.
Thank you, Sandra.
Thank you.
And we'll make sure those last couple questions get answered. Thanks everybody for a great session. Great job.
Thank you. Bye-bye, everyone.
Thank you. Bye.
Bye.